Welcome to Bio 2. This is chapter 24, which is our first chapter of Bio 2. Bio 2 is basically the second half of the book. So chapter 24 is largely about the origins of life, a little reminder about the three domains of life, and then some information on prokaryotic organisms. This whole first unit is really about diversity of life. So let's jump into how life began. Most scientists propose 4 overlapping stages that had to occur for life to begin. So we needed organic monomers to arise. What's a monomer again? Remember, the root word mono means one. So a monomer is a simple organic molecule like an amino acid or a monosaccharide, something like that is an organic monomer, or a nucleotide would be an organic monomer. Then organic polymers needed to come about. Then we needed to have boundaries or kind of early protocells. And then we needed cellular properties like self-replication. So there's several different ideas about how organic monomers could have come about. Again, we needed nucleotides and amino acids. Those are critically necessary and important for life. Early Earth may have been more conducive to the spontaneous formation of organic molecules. There may have been kind of a prebiotic or abiotic synthesis because there was not much oxygen, so there's kind of this prebiotic soup that was around. There's several different hypotheses on where and how organic molecules originated, and we'll go over these on the next couple slides here, but there's the reducing atmosphere hypothesis, the extraterrestrial hypothesis, the deep sea vent hypothesis, and also volcanic eruptions could be responsible for organic monomer formation. So on that, organic monomers arising, one possible explanation is the reducing atmosphere hypothesis. So way back in 1953, a couple of researchers were able to simulate early Earth's atmosphere in this sort of complex glass apparatus pictured here on this slide. And in that, they were able to synthesize amino acids, monosaccharides, lipids, and nitrogenous bases, so important monomers necessary for life. We don't do this experiment in undergraduate biology classes, though, because it's really important that you remove the atmospheric air, because atmospheric air has more oxygen in it, and if we didn't remove that and then we sparked inside of the glassware, that would cause the glassware to blow up. So it's a relatively dangerous experiment for undergraduate biology students to perform, but it has been repeated multiple times, and they've even reanalyzed the material that was produced way back in 1953 with more modern equipment, and still found these organic monomers. Another possible explanation for how we got organic monomers is the extraterrestrial hypothesis. Honestly, this one's not really my favorite, and I don't think I'm alone in that. So the idea with this one would be that meteorites brought organic carbon to Earth, including things like amino acids and nucleic acid bases. Opponents to this would argue that most of these molecules would be destroyed in the intense heating and collisions. And another problem I personally have with this hypothesis is, like, it still doesn't explain like where those monomers came from. So perhaps an unsatisfying explanation for organic monomers arising is the extraterrestrial hypothesis. Another possible explanation for organic monomers arising is that there's biologically important molecules have the ability to form in a temperature gradient between extremely hot vent water and cold ocean water. In 1988, researchers demonstrated the synthesis of ammonia from nitrogen gas at the gradient between seawater and materials emitted at hydrothermal vents. This ammonia is necessary for nucleic acids and amino acids to form. In present day, still, there's complex biological communities found at these deep sea vents, and they derive energy from chemicals in the vent. So they do chemosynthesis. That's kind of the base of those communities, rather than photosynthesis. There's some evidence that suggests that early Earth's atmosphere was neither reducing nor oxidizing, and so volcanic eruptions themselves could be responsible for organic monomer formation. So once we've got monomers, we could link them together and form organic polymers. So small organic molecules polymerize when they're concentrated on things like hot sand, clay, or rock. So formation of nucleic acid polymers and polypeptides, so that's chains of amino acids, were seen to link together way back in 1951. Cations bind the nucleotides to the negative surface of the clay, and that really promotes the bond formation between the phosphate and the ribose sugar of those nucleic acids. There's been experimentally less success in solution because hydrolysis competes with polymerization. What's hydrolysis again? Remember, hydrolysis is when we add a water molecule in and break a polymer apart, and polymerization is building the polymer. So basically that sentence is saying that breakdown competes with building in water. But clay surfaces seem to be important for organic polymer formation. As far as formation of boundaries or formation of protocells, so cells as we know them today have cell membranes, and in water, lipids and other organic molecules can spontaneously form vesicles with a lipid bilayer, right? Remember, the present day cell membrane is a phospholipid bilayer, so 2 layers of phospholipids. Protocells may have been fluid-filled vesicles with a membrane-like structure, and adding this clay that's found near volcanic eruptions can actually increase the rate of vesicle formation. And vesicles exhibit simple reproduction and metabolism and maintain an internal chemical environment. So that's an important property for early life. As far as cellular properties go, a cell needs to be able to store information, it needs to be able to replicate, and it needs to have enzymatic functions, right, to control the rate of chemical reactions inside of this cell. So many favor RNA as the first macromolecule because it can do all of those things. DNA and proteins can't do all 3 functions. So DNA is not a good catalyst at all, and we haven't found any self-replicating proteins. So we think the first biologically important macromolecule was likely RNA. As far as fossil evidence of early life goes, don't get too hung up on memorizing like the fossil record. I think it kind of depends on who your biology teacher is, how much detail you'll go into on that sort of thing. But I just kind of want to broadly introduce it to you, and we'll focus more on diversity of life and characteristics of present day life. So Earth formed about 4.6 billion years ago, and some of the earliest prokaryotic cells lived in dense mats, while others were free-floating individual cells. And many of the oldest fossils are stromatolites, which are these layered rocks that formed from the activities of prokaryotic organisms up to 3.5 billion years ago. And you can see an image of that on this slide. But we've also got ancient fossils of individual prokaryotic cells that have been discovered. For example, there's pictures on this slide of non-photosynthetic and photosynthetic individual prokaryotic cells as well. So let's review. So what are 2 differences between prokaryotic and eukaryotic cells? If you want to pause it and think about it, you can think about it before you hear my response. So remember, prokaryotic cells don't have a nucleus. Eukaryotic cells do have a nucleus. That's one difference. Another difference, prokaryotic cells are smaller than eukaryotic cells. A third difference would be that prokaryotic cells don't have membrane-bound organelles like eukaryotic cells do. Let's go ahead and get to number 2. Circle the domains of life here. So domains of life. Does anybody remember how many domains of life there are? There are 3 domains of life. What domain are we in? We are in eukarya. So that's a domain. And then there's, people always want to say prokarya, but that's not a domain. There's prokaryotic cells and eukaryotic cells, but there's actually 2 prokaryotic domains. So there's archaea and bacteria. So the 3 domains of life are bacteria, archaea, and eukarya. So to kind of compare those 3 domains of life, we've got bacteria, archaea, and eukarya in this table. Bacteria and archaea are both prokaryotic, so they're not going to have a nuclear envelope, and they're not going to have membrane enclosed organelles. Eukaryotic organisms have both of those things. Bacteria have peptidoglycan in their cell wall, but archaea and eukarya do not have those things. As far as the membrane lipids go, bacteria and eukarya have similar cell membranes. Archaea have different cell membranes, and we'll talk about that more coming up later. Some of that has to do with their ability to survive in extreme environments. Does anybody remember what RNA polymerase does? I guess it's probably just me and one person, but okay. Do you remember what RNA polymerase does? So RNA polymerase, it builds the RNA polymer, right? What does that mean? So RNA polymerase reads DNA to make RNA. Bacteria have one kind of RNA polymerase, but archaea and eukarya have multiple kinds. As far as growth at temperatures higher than 100 degrees Celsius, that's really hot. Bacteria can't do that. Eukaryotic organisms can't do that, but some species of archaea can do that. But wait, there's more. So this table continues on another slide. Again, we've got bacteria, archaea, and eukarya, and we're comparing them. Introns in genes. Do you remember what an intron is? So remember, there's introns and exons. Introns are those intervening sequences that don't make it into the mature mRNA. So we really wouldn't expect bacteria to have them. Archaea have some of them. Eukarya, it's pretty common to have introns in genes. Response to antibiotics. So bacteria, their growth is inhibited by antibiotics typically, but archaea and eukarya, their growth is typically not inhibited by antibiotics. Now, there's, we could have an argument about this, and we could have the whole dose makes the poison conversation, but we're talking generally speaking. So generally speaking, bacteria, their growth is reduced in response to antibiotics, but archaea and eukarya not reduced. Histones associated with DNA. Does anybody remember what a histone is? So histone, remember, is that protein that the DNA winds around to package the DNA. So not present in bacteria, in some species of archaea, and in many eukaryotic organisms, it is present. So not bacteria, but in archaea and eukarya. And then circular chromosomes. Typically, bacteria have circular chromosomes and archaea have circular chromosomes, but eukaryotic organisms have linear chromosomes. Broadly, for prokaryotic organisms, we have bacteria and archaea. They are both unicellular, although sometimes they kind of blur the lines with this because some species form colonies, and we'll see later on there's some metabolic cooperation as well. But broadly, we would consider these to be small unicellular organisms. So again, prokaryotic cells are smaller than eukaryotic cells. I'm not going to have you memorize the exact micrometers, but prokaryotic cells are much smaller than eukaryotic cells. Prokaryotic cells are going to tend to have cell walls. They also are going to have a cell membrane. Those cell walls help prevent osmotic lysis. What osmotic lysis is when water rushes into the cell and causes it to rupture. Remember, osmosis is the movement of water across a semipermeable membrane, and lysis is rupture. So there's the variety of shapes of prokaryotic organisms, and if you take a microbiology class, you will get into this in so much more detail. We're just kind of giving you a broad overview here, but The spherical shape is cocci or coccus. The rod shape is bacilli or bacillus, and there's several different kinds of sort of spiral-shaped cells. There's spirochetes, spirillii or spirillum, and vibros, which is more kind of comma-shaped. Again, there's two prokaryotic domains. There's bacteria and archaea. There's a lot more phyla of bacteria than archaea. There's 50 or more bacterial phyla and only 5 archaean phyla. The cell membrane lipids in bacteria and archaea are different. I don't expect you to memorize like ester-linked unbranched fatty acids and ether-linked branched isopurine chains, but I do want you to know that structurally the membranes are different. The lipids present in archaea's membrane allows some of them to be extremophiles or live in extreme environments. Bacteria have cell walls with peptidoglycan in them. Archaea do not have peptidoglycan. They often have a protein-based cell wall. There are many bacteria in symbiotic relationships with eukaryotic organisms. They could be mutualist, they could be in commensal relationships, they could be parasites, and kind of so on and so forth. There, many bacteria favor moderate conditions. There's limited extremophiles here. With archaea, they're sort of known for being extremophiles, but many of them do live in moderate conditions. And archaea has features similar to the eukaryotic nucleus and cytoplasm, which we covered in those tables before. Like archaea has histone proteins, similar ribosomal proteins, and similar RNA polymerases. Both domains, though, have diverse metabolic features and habitats, and we're going to kind of introduce you to some of the diversity of habitat and metabolism coming up here. When we talk about bacteria, oftentimes you might hear gram-positive or gram-negative bacteria. Gram staining is clinically important because it gives us some information about the bacteria. And gram-positive bacteria are vulnerable to penicillin because it actually interferes with their cell wall synthesis, while gram-negative bacteria are resistant to penicillin. So gram-positive bacteria have a thick peptidoglycan layer. So in the gram staining process, they retain this purple dye, so they tend to be stained this dark purple color. Gram-negative bacteria, on the other hand, have a thin peptidoglycan layer, and they have an outer envelope as well. So they do not retain the purple stain. Instead, they retain a pink stain. So you can see at the bottom of the slide here, there's gram-positive bacteria that are kind of this purple color, and gram-negative bacteria that are this pinkish color. So let's review which bacterial shape is represented in this picture here. If you can't see the picture, because you're like on the treadmill or something, it's the rod-shaped bacterium. So that's going to be bacillus is the shape that is represented there. For the next part, we've got 2 pictures, and it says which image would be gram-positive and why. So again, pause if you want a moment to think about this. But if we look at the pictures, we can see there is a thick peptidoglycan layer on this one and a thin layer on the other one. The second picture also has this outer membrane. So our first one is going to be gram-positive, and the second one is gram-negative. And again, this thick peptidoglycan layer is really letting us know that this is the gram-positive slide. So then some cell surface extensions of prokaryotic cells. Prokaryotic cells can have flagella. These flagella differ from eukaryotic flagella structurally and functionally. Flagella likely evolved independently in all 3 domains. Like flagella are very beneficial to organisms. Bacteria can also have, prokaryotic cells in general can also have pili, which are thread-like structures that can allow the cells to kind of twitch or glide. These pili can also be important in gene transfer and disease processes. Maybe you've heard of conjugation pili or sex pili before. And there's also fimbriae, which are not pictured here, but they're hair-like appendages that allow the cells to stick to substrate or to each other. As far as internal organization of prokaryotic cells go, prokaryotic cells lack complex compartmentalization. It's not that they don't have any sort of specialized structures. Some of them have specialized membranes that perform metabolic functions, like in the picture below, there's an aerobic prokaryotic organism with a respiratory membrane that does cell respiration to make ATP. There's a photosynthetic prokaryotic organism pictured here with thylakoid membranes to do photosynthesis. As far as the genetic material in prokaryotic cells go, the prokaryotic genome is smaller than the eukaryotic genome. Typically, they have a single circular chromosome found in a region that's called the nucleoid region. It's not enclosed in a membranous nucleus like eukaryotic cells have. They can also have smaller accessory rings of DNA that are called plasmids. To survive unfavorable conditions, some bacterial cells produce something called an endospore. The endospore has DNA and a tough protein coat, and these endospores are tough to destroy. So they need several minutes of a 10% bleach solution. And let's be serious now. When you wipe off like a countertop or something like that, if you feel like you need to get bleach, are you waiting several minutes? Most likely not. So if you think you are trying to remove bacteria with a chemical, you should definitely read your label to make sure you're following the instructions. Lots of times you need to have a certain time period that the chemical needs to stay moist or wet in order to actually remove bacteria. You can also kill endospores with autoclaves or ionizing radiation. An example of endospore forming bacteria would be Clostridium botulinum and Clostridium tetani. So if you've heard of, you probably have like had a tetanus shot maybe. So tetanus, the disease state causes lockjaw. It causes your muscles to contract uncontrollably. Your diaphragm is a muscle. If you can't control that, you're not breathing. That's a bad thing. Botulinum toxin, if you've heard of Botox before, like again, the whole dose makes the poison. So a small amount of botulinum toxin in Botox could be useful to, there's different things that they can do besides just like Botox for cosmetic reasons. But basically it causes a small amount of paralysis with a controlled injection of that. Whereas if you get too much of that, again, your muscles don't respond to stimuli. You need your diaphragm to breathe and so on and so forth there. So this one's kind of a preview review situation. You may remember autotroph and heterotroph from Bio 1. Like a photosynthetic organism is an autotrophic organism, right? Because they are able to produce their own organic molecules with light energy. And like a heterotrophic organism, that's what we would be. We need to have preformed organic molecules, and we get our energy and our carbon from those organic molecules. So the root word for light is photo, right? So if we look at our first one here, the energy source is light and the carbon source is carbon dioxide or bicarbonate ion. This would include photosynthetic prokaryotic organisms, plants, and algae. So if we put that root word for light and the root word for their energy source together, their carbon source together, we get, if it'll do it, we get photoautotroph. So for our next example here, we have the energy source is light. So what's the root word for light? Photo. You got it. Their carbon source is preformed organic molecules. So that would be a photoheterotroph. Our next one, the energy source is chemicals and the carbon source is carbon dioxide. So the root word for when their energy source is chemicals is going to be chemo. So this is a chemoautotroph. So like those organisms at kind of the base of the food chain and those hydrothermal vents in the oceans would be chemoautotrophs. And then if the energy source is organic compounds and the carbon source is organic compounds, that's going to be a chemoheterotroph. So we would be a chemoheterotroph. There's many protists and prokaryotic organisms and fungi that are also chemoheterotrophs. So let's review. What are two structures that help bacteria move? That would be pili and flagella. Number 7 is kind of a review from Bio 1. Which of these describes gene transfer between different species? That's going to be horizontal gene transfer. A vertical gene transfer is from parent to offspring. And then number 8, what specialized dormant structure do some species of bacteria form to survive extreme environmental conditions like heat, desiccation, and radiation? That would be endospores. Okay, the writing is not great, but endospores. We can also classify prokaryotic organisms by their oxygen response. So there are obligate aerobes, obligate anaerobes, and facultative anaerobes. If you are obligated to do something, what does that mean? It means you have to do it. And then we have aerobic and anaerobic. The root word an means not or without. The root word aero means, technically it means with air, like molecular oxygen O2. So obligate aerobes will require O2 because it is the final electron acceptor in the electron transport chain of cellular respiration. Obligate anaerobes cannot tolerate oxygen. They can't deal with hydrogen peroxide that's built up in their cells. So they cannot be in an environment with oxygen. Then we have facultative anaerobes, and they could go either way. They could use oxygen or not. They're capable of either fermentation or anaerobic respiration, and also aerobic respiration, but they're not poisoned by oxygen. There's also diversity in nitrogen metabolism and some cases of metabolic cooperation between bacteria. So nitrogen is needed for amino acids and nucleic acids. Remember, amino acids link together to form proteins. Nucleic acids build DNA and RNA. So arguably those are both important things. Some prokaryotic organisms can do something called nitrogen fixation, where they can convert atmospheric nitrogen gas to ammonia, which can be used in the production of organic molecules. Some bacteria have cooperation, metabolic cooperation. So, for example, in some cyanobacteria, they have photosynthetic cells and nitrogen-fixing cells that are called heterocysts or heterocytes, and these cells exchange products. They have to cooperate because the photosynthetic cells produce oxygen, and that would disrupt nitrogen fixation, but the photosynthetic cells benefit from the ammonia that's produced in nitrogen fixation. So you can see in this slide here, there's green photosynthetic cells and those yellow heterocysts, which are the nitrogen fixing cells. Biofilms are another kind of cooperation. This is when prokaryotic organisms can create kind of the surface coating colony. The cells actually start to generate an extracellular matrix, so they secrete material outside of the cells that link the cells together. This forms on virtually every non-shedding surface and non-sterile, humid, or water-based environments. Dental plaques are an example of a biofilm. If you've ever had a clogged pipe, there's often biofilms in there. Biofilms increase drag in the shipping industry. If you've ever tried to walk across a stream and you slipped on a rock, that rock was slippery because there was a biofilm on it. There's problems with biofilms on implanted medical devices, and also they lead to difficulty in sanitizing surfaces that are required for food preparation. So how do prokaryotic organisms reproduce? Well, they reproduce by binary fission. This may be a review from Bio 1 for you, but basically binary fission is relatively simple because there's a single circular chromosome. That chromosome replicates, it splits into two, it produces two identical chromosomes, and then the cell itself splits into two, and that produces two identical cells. So prokaryotic organisms can quickly divide because they have small cells. Binary fission is a relatively simple process, and they have short generation times. So there's not a long time period where they have to be around before they can reproduce. So even though prokaryotic organisms make identical copies of themselves, there is still considerable genetic variation. Since they reproduce so rapidly, even though like the mutation rate in binary fission is low, since they reproduce so rapidly, mutations can accumulate rapidly. Also, prokaryotic organisms can do genetic recombination, where they combine DNA from two sources, which contributes to diversity. You may remember from Bio 1, they can do transformation, transduction, and conjugation as horizontal gene transfer, which is genetic recombination. So if you don't remember transformation, conjugation, and transduction, transformation is when prokaryotic cells can take up and incorporate DNA from the surrounding environment into their bodies. Conjugation is when genetic material in a plasmid is transferred by way of sex pilus or conjugation pilus from one cell to another. And transduction is when phage, which is a virus that infects bacteria, transfers the genetic material from one cell to another. So those are all examples of horizontal gene transfer between bacteria. Okay, so let's review. Let's match the picture to the terms. So in A, right, this bacterium has released genetic material into the environment, and this other bacterium is picking it up there. So what's it called when the bacteria picks up genetic material from the environment? That is transformation. In B, this bacterium, this bacterium is giving genetic material to the other bacterium through this kind of tube here. That tube is a pilus, so this one is conjugation. And then this little spaceship looking doodad is a virus, and it came from this cell to that cell. So we're left with transduction, and that is correct for C. And then bacteria reproduce asexually through what? What is that called? It's binary fission. My handwriting is so terrible. There's actually a huge amount of diversity in prokaryotic organisms. If you recall, again, in the 3-domain system, 2 of those 3 domains are actually prokaryotic organisms. They inhabit every environment known to support life and have every major mode of nutrition. And we have looked at revising the prokaryotic phylogeny because we've gotten more information with molecular techniques. So remember, bacteria and archaea are both prokaryotic groups, but they're not a monophyletic grouping. Archaea are actually more closely related to eukaryotic organisms than they are bacteria. And honestly, horizontal gene transfer really obscures and complicates these phylogenetic relationships. So here's kind of a brief overview of some bacterial groups. There's 16,000 known species of bacteria, but estimates range from 700,000 to 1.4 million species of bacteria. They're very, very numerous. So 2 groups that I want you to memorize are proteobacteria and cyanobacteria. You might think, oh, I could memorize all of these other groups as well, but there's plenty of memorization in this first unit, so just memorize proteobacteria and cyanobacteria. This is likely because I'm biased towards eukaryotic organisms, and proteobacteria are gram-negative bacteria, and they have diverse metabolic and nutritional modes, but mitochondria are thought to have evolved from alpha proteobacteria. Cyanobacteria are gram-negative photoautotrophs that generate oxygen through photosynthesis, and plastids like chloroplast evolve from cyanobacteria. Some other bacterial groups which you don't need to memorize are chlamydias, which are gram-negative disease-causing parasites that require animal hosts. Spirochetes, which are helical, so twisty, shaped like a DNA helix, right? Gram-negative heterotrophs. Many species are free-living, but some are parasitic. The causative agent of syphilis and the causative agent of Lyme disease are both spirochetes. Then, of course, there's gram-positive bacteria, which are a pretty diverse group, but some include soil decomposers and pathogenic bacteria. So, again, only memorize proteobacteria and cyanobacteria. So, I don't know if I made it clear enough, but I only want you to memorize proteobacteria and cyanobacteria. This slide has a couple of the different kinds of proteobacteria. So, I told you alpha proteobacteria are important because of the relationship with present-day mitochondria, but there's other kinds of proteobacteria as well. H. pylori is responsible for many stomach ulcers, and if you're interested in microbiology, that's kind of an interesting story to read about how that was discovered. And then our friends, the cyanobacteria, on the other side of the slide are photosynthetic. Here's other bacterial groups. Make note of the little graph on the left-hand side that says known species of bacteria 16,000, and look at how much more of the circle is not in that little tiny yellow edge there. So more species of bacteria are undiscovered and unnamed than we actually have discovered and named. So that brings us to archaea. Archaeas share some traits in common with bacteria and others with eukaryotic organisms. They're most famous for being extremophiles. They're not all extremophiles, but the ones that are capable of living in extreme environments are extremophiles. For example, halophiles are able to tolerate or require a highly saline or salty environment. Extreme thermophiles have adaptations that stabilize their DNA and proteins and allow them to thrive in extreme heat. There's other extremophiles like alkalophiles and osmophiles and so on and so forth. And if you take a microbiology class, you may get into that. So prokaryotic organisms are very important to life. So they are important for functional biogeochemical cycles, which we'll talk a little bit more about when we get to Unit 5. They form close relationships like mutualism, where both organisms benefit, commensalism, where one organism benefits and the other doesn't. They're not helped or harmed, and parasitism, which I think when people think bacteria, they're probably thinking pathogenic bacteria or disease-causing bacteria. Bacteria are also, or prokaryotic organisms in general, are also important for bioremediation and research and technology. So again, I think people are, pretty aware of pathogenic bacteria. Prokaryotic organisms cause about half of all human diseases. For example, Lyme disease is caused by a bacterium that's carried by ticks. Pathogenic prokaryotic organisms typically cause disease by releasing exotoxins or endotoxins. Exo means outside of, endo, inside of. So exotoxins are secreted and cause disease even if the prokaryotic organism that produced them are no longer present. Endotoxins, on the other hand, are released only when the bacteria die and their cell walls break down. There's some bacteria that are in mutualistic relationships with another species. So our intestines are home to about 500 to 1,000 species of bacteria, and many of these help break down food that's undigested by our intestines, and some of them even help synthesize vitamins. Those hydrothermal vent communities depend on chemoautotrophic bacteria to be kind of the base of that ecosystem down there. And then here's just kind of a fun example. There's a bioluminescent bacteria on this flashlight fish that help attract the fish's prey. In research and technology, maybe you remember from Bio 1 hearing about gene cloning. E. coli is frequently used for gene cloning. Maybe you remember PCR, polymerase chain reaction, where that requires a heat-stable DNA polymerase. That heat-stable DNA polymerase is from a prokaryotic organism. If you remember the CRISPR-Cas9 system, which is used for altering genomes, that originally developed in prokaryotic organism. That wasn't just us being super clever and figuring that out on our own. It's a naturally occurring system in prokaryotic organisms. And then bioremediation is when organisms can remove pollutants from the environment. The agar dishes on the right-hand side there show degradation of heavy crude oil within 36 hours of incubation with a certain bacterium. I cited the journal article below that if you're interested in that. But there are prokaryotic organisms that are capable of remediating for oil spills, digesting plastics, paint thinner, helping clean wastewater, and removing heavy metals. If you're super interested in that, there's a microbe wiki with lots of lists of microorganisms and which specific things they are good for remediating. So we made it to the end. So I like to kind of review at the end, but it's not, a total thorough, perfect review. So make sure you check your reading guide and, go over your notes again. But we talked about the 4 overlapping stages that were required to produce life, right? So we had to have organic monomers form, polymers form, boundaries, and cellular properties. The first genetic material was likely RNA because RNA can act as a catalyst, can self-replicate, and can store information. We talked about common prokaryotic shapes. So the rod shape is bacillus. The circular shape, like spherical shape, is coccyx or cocci. And then the squiggly shapes, we've got spirillum, spirochetes, and vibros is a comma shape. Gram-positive retain purple dye, have a thick peptidoglycan layer. Gram-negative bacteria have a thin peptidoglycan layer and retain a pink dye. Prokaryotic organisms have cell walls. Bacteria have peptidoglycan. Archaea tend to be more protein-based. Some bacteria can form endospores to survive unfavorable conditions. Pili could be for movement or for conjugation, in exchange of genetic material. Then fimbriae attach bacteria to the substrate or each other, and flagella are for movement. Prokaryotic DNA is contained in a single circular chromosome that is found in the nucleoid region. They also sometimes have smaller accessory rings of DNA called plasmids. There are many types of metabolisms in prokaryotic organisms. They can do aerobic and anaerobic respiration, and they can also, some of them can do nitrogen fixation. We talked about cooperation between colonies, particularly with the cyanobacteria, with the photosynthetic cells and the nitrogen fixing cells, those nitrogen fixing cells and those heterocysts. Bacteria reproduce by binary fission, which is an asexual kind of reproduction, but there's genetic diversity because they can do horizontal gene transfer through conjugation, transformation, and transduction. The 3 domains of life are bacteria, archaea, and eukarya. So 2 of those 3 domains are prokaryotic. Archaea are sometimes extremophiles, meaning they can live in extreme environments, and 2 specific kinds of extremophiles are halophiles, which live in salty environments, and thermophiles, which live in high temperature environments. That should say cyanobacteria, and on your notes that'll be fixed, but that's a C. Yep. So then the 2 bacterial groups I want you to memorize are proteobacteria and cyanobacteria. Proteobacteria in particular because alpha proteobacteria are genetically similar to modern day mitochondria, and cyanobacteria because they are similar to plastids like chloroplasts. And then if I ever ask you an importance of question on the exam, I'm trying to have you show me that you paid attention and that you made some connection. So prokaryotic organisms are important for many reasons, including that they're they are pathogens. Some of them are in mutualistic relationships. They're important for functioning biogeochemical cycles. They're important for research. That's not a comprehensive list, but a couple examples there. So we made it through chapter 24.